Hello, hello students. This is Dr. Faizan Mirza.
In this video, I'm covering the details of t-test. So what actually is t-test? T-test, whenever the t-test question comes, the formula will be given to you. I've taken this question from a P4 that came in October, November 2018, variant 41. And this question actually they dealt with meiosis and some details of the general details of other parts of your syllabus, including the genetic technology and selection evolution, etc. And then it came to t-test. So let's start with the question here. So generally, they are asking initially meiosis, one process that contributes to genetic variation, state precisely the stage at which meiosis, where single chromosomes line up on the equator. So now you know this question is basically a factual question. So you can just recall that it is metaphase 2, and you can write the answer. It can't be metaphase 1 because they are saying single chromosomes here. Then the next part outline the events that take place during anaphase 1 of meiosis. So anaphase 1 of meiosis, so you can just recall again the facts of anaphase 1. You can state that microtubules, spindle fiber, they start to shorten, the chromosomes start to get pulled towards the centrioles, and the centromere are pulled first. We need to make sure that we highlight the point that the centromeres are pulled first, and because of the centromeres being pulled apart, the homologous chromosomes start to separate out. If it's a 2-mark question, then obviously you have to have 2 marking points in the question. And since the question deals with the command word outline, so you just need to just give answer in brevity. The answer needs to be brief. The answer does not need to have any extreme details or reasons for why it's happening and what's happening. The next part they say describe how crossing over during meiosis lead to genetic variation. So you'd again recall the facts of crossing over. So what are the facts of crossing over? We know crossing over occurs on the non-sister chromatids, which lie close together, and they exchange their segments. The alleles that are present on these segments, they get swapped to the region where the crossing over occurs, called chiasma. This results in production of the new combination of alleles. So yes, you have to make sure that you mentioned it's occurring in the between the non-sister chromatids, which are lying close to one another. Now the next question here, and the next part of the question moves to another part of your syllabus. They bring mutation in question. So the first thing that you have to do when you are reading a question is to identify which topic the question deals with. So mutation also called genetic variation. So you know the question deals with mutation, the question deals with genetic variation. Some populations of water hemp, there's an organism water hemp, there's the first letter and the second letter. This is how we name our certain organism. The first word here represents the genus of the organism. The second word here represents the species. They say that it evolved herbicide resistance as a result of mutation. We know most of the resistance comes as a result of mutation. Any new allele comes because of mutation. So we know the concept of a new allele and mutation. They both go hand in hand, and we have studied this in selection and evolution in detail. So they say that this mutation is actually a problem for farmers where this water hemp grows in crop fields because it lowers the productivity. So we know that water hemp actually is actually harming the crop because it competes with the crop for the same resources. We can just figure this out from this part. They say further, 2 populations of water hemp were tested for resistance to herbicide mesotrione. Now there's a herbicide mesotrione. The mesotrione is actually killing water hemp. And this is used by farmers to just make sure that water hemp gets killed so the crops survive. Now they are saying that one was a population known to be resistant, which is controlled. Another population was a test whose resistance was unknown. So there are 2 populations. Now 2 populations mean that they are comparing 2 sites. When 2 sites are being compared, it means we are having 2 sets of data. One data is for the resistant control, which can degrade mesotrione, and the other is a test population whose resistance is unknown. We do not know what the resistance is. The further 3 bullets give you the details of this experiment. They say leaves were removed and immersed in a radioactively labeled solution of mesotrione, which means mesotrione should be absorbed and should be taken up. The leaves will become radioactive. Resistant leaves are then able to degrade mesotrione by metabolism. Why would be able to degrade mesotrione? Because we know that because of this mutation would have given a new allele. The new allele would then have caused the resistant leaf to be able to degrade mesotrione by metabolism. Degrading because probably it might just have some kind of enzyme which can actually just deactivate mesotrione. The time for 50% absorbed mesotrione to degrade was calculated and measured radioactivity of the leaves. So the results are shown to you in table 3.1. The population of water hemp test and control. We know the control is the resistance one, so we can just annotate that resistance actually is present in control. Mean time for 50% of this mesotrione to degrade, we can see that the mesotrione is getting degraded in just 10.1 hours in the control, which is having resistance allele, whereas the test whose resistance is unknown, it is taking 27.5 hours to degrade 50% of the mesotrione. So obviously this we know it is taking a longer time, and this is taking a shorter time. So obviously when it's taking a shorter time, it means that this is more evolved in getting the mesotrione degraded. The standard deviations are given to you. What's the standard deviation? Standard deviation, you need to again learn the fact that standard deviations actually tells you the spread of data, and the spread of data actually around the mean value. And the standard deviation value, if the standard deviation value is larger, it means that the data is more variable, and there's that it's more varied, not variable. The data is more varied, and it is less reliable. I have covered standard deviation in detail in one of my videos, so I'm not going details of that right now. So how do you mention the standard deviation? The standard deviation can be mentioned with mean plus minus standard deviation. For example, if the mean is 27.5 and standard deviation is 4.75, so we can just mention this, or this can be written as 27.5 plus minus 4.75, which means that the value above mean could be 4.75, or below mean it could be 4.75 as well. For example, 10.1, the value of standard deviation is 2.34, which means it can be 2.34 above or below 10.1. So we can see, if you compare the two standard deviation values, this actually is showing a less varied population, because the standard deviation value is shorter or smaller. Here the standard deviation value is larger, so that obviously means that this is a more varied data, or data is less reliable. Next, they're asking you explain how the results in table 3.1 show the two populations differ in the resistance to mesotrione. So you can just say that obviously what we need to just give reason here. So we'll just mention that test is taking longer, the control is quaking, is actually degrading it in less time. We can just mention test leaf group takes longer time to degrade mesotrione, so they are less resistant. And we know if they are less resistant, so why are they less resistant? They are less resistant because they are having an allele which came because of mutation. The standard deviation of the test is large. This group is more varied than control, more variable than control. Explain why this example of genetic variation is important for natural selection in water hemp population. So why is this example important for natural selection? We know that the hemp, which is resistant, it is more evolved. It is more evolved. Why is it more evolved than the wild type? Because it is able to grow even in a region and where the mesotrione herbicide is being used. So what's its outcome in the natural selection? Yes, they can adapt and evolve. This helps in their survival. The native plants would have a selective advantage in areas where mesotrione is used, and they would survive in. So yes, we have just linked the details of mesotrione and the fact that we know already about natural selection, that the better adapted ones survive. Then they're saying farmers can send in sample of water hemp from their fields for a laboratory to be tested for resistance to mesotrione or other herbicide, suggests the benefit to this to farmers. So why a farmer would send the samples of the leaves to a lab? Obviously, because the farmer would want the leaves to be tested for the resistance of various herbicide which he or she is using. Now, in that case, the farmer would already know from the lab results which is the most suitable of the herbicide that he or she should use. This is exactly equivalent to the culture sensitivity test that we carry out for testing any microorganism for a particular antibiotic. Because, for example, if a person is suffering from an infection in blood or in urine, and it is not being treated by any broad spectrum antibiotics, so you send the urine sample for culture sensitivity or the blood sample for the culture sensitivity, and the culture sensitivity test then actually tells the doctor through the experiments which antibiotics are most effective and which ones are least effective. So this exactly is the similar case, that the farmers can choose most suitable herbicide that is most effective, and less money would be wasted in trying other herbicide because trial and error is not being done. The crop yield would stay high here. Now the next part of the question goes to t-test. Actually, this is why I am discussing this question in detail with you. Now the question begins with the null hypothesis. It states that there is no significant difference between the mean times of 50% absorbed mesotrione to degrade in two populations. Whenever we state the null hypothesis, they may ask you to state the null hypothesis. We'll always use there is no significant difference between the mean. This statement must be there when you are discussing the null hypothesis for a t-test, because a t-test is a test in which mean is being compared between the two groups of population or two sets of data. Otherwise, whenever we are stating null hypothesis for any other statistical test, for example, chi-squared, Spearman, or Pearson, we need to keep in mind that we'll again keep on mentioning there is no significant difference between whatever we are testing. And for example, if it's correlation, if it's mean, if it's observed and expected, whatever it is, we just mention it next, and then we mention the variables. So in this case, this was already mentioned that there is no significant difference in population between the population to degrade the mesotrione. A t-test was carried out to compare the two means. The critical value of t-test at p 0.05 was significance level was 2.23. So this is the critical value. We know in a statistical test, we already either are given a critical value or we compare our statistical calculated value with the critical value. So this t-test formula is given to you. T-test, when do we use t-test? Basically, this again is fact that you need to keep in mind where t-test is used. T-test is used when data is continuous. Continuous data, whenever the data is continuous, you will be able to calculate mean. So the data is continuous, yes, you will apply t-test. It is used to compare the means of two sets of data and to see whether there is a significant difference in the mean or not. So we use t-test for that. And now you know we already have a critical value. We'll calculate a t value here and we'll compare the two values together. We'll compare our calculated t value with the critical value 2.23. The formula given to for you for the t-test is this formula is given to you. Let's just decode this formula. So this formula actually is x bar 1 minus x bar 2. X bar represents mean. So since there are two sets of data, wherever one is mentioned, it means this is the first set of data. In this case, this can be control or the unknown resistance, and this other can be the control, which actually was having resistance. So we have two sets of data set 1 and data set 2. So x bar represents the mean, s represents the standard deviation, n represents the number of readings which were taken. So the number of readings taken for both is 6 each. This is already mentioned to us. What will we do? We'll just start to put these values in their appropriate places and we'll solve it for the test. Now, how the values are coming? X bar 1, X bar 2. Where did we give? Where were these stated? They were stated already in the previous table 3.1. Test mean was given to you. Control mean was given to you together with the standard deviation. So we'll just mention these standard deviation and the mean in the respective positions. So this is the mean and the standard deviation, the mean and the standard deviation. And we know that the formula asks for standard deviation to be squared. So we'll just square the standard deviation. We'll square the standard deviation as well. We'll divide each by 6. Where did 6 come from? 6 was already given as the number of readings here. Now, when we'll just calculate, we'll just solve it as a mathematical equation. Our answer comes out to be 7.98. Now, we know that the critical value is 2.23 and the calculated t-test value is 7.98. What will we do? We'll start to compare the two values together. This is what we do in statistics. We compare the calculated value with the critical value and we see whether our calculated value is larger or smaller than the critical value. In this case, we know that our value of t-test 7.98 is way larger than the critical value 2.23. Because after 2.23, you will have 3, 4, 5, 6, and 7, and only then you will have 7.98 coming. So our critical value is way smaller and our calculated t value is way larger. So what this leads us to? So they are asking that your calculated value of t to explain whether the null hypothesis would be accepted or rejected. So whenever the calculated value of t-test is larger than the critical value. The critical value given to us was 2.23. Our t-test value was 7.98. So whenever the t-test value is larger than the critical value, we reject the null hypothesis. And we say that the difference is significant and it's not by chance. So we reject the null hypothesis. The explanation would be that calculated t-value is higher than the critical value of 2.23. Probability result means by chance for 0.05. There is a significant difference in the means of two groups of data shown to you. This 0.05, p less than 0.05 represents 1 in 20 chance of error, or you can say 95% confidence or 5% chance of error. So this is quite similar to what we do in chi-squared as well. In chi-squared test, we actually calculate the chi-squared value and we compare it against a critical value. So this is what I wanted to discuss with you regarding t-test. That's it from my side. Thank you so much.